Nivedita Joshi is here by taking a new video on fertilization and implantation. In the previous video, already you have gone through the topic spermatogenesis, that is oogenesis, menstrual cycle and all. Now after that we have a process fertilization we will study in the human beings. So as you have gone through, fertilization means it is the fusion of male gamete with the female gamete and the resulting cell is zygote. Means the gametes are haploid and the zygote is diploid. Means to the haploid to the gametes we can represent them as N, and to the zygote we can by 2N. So fertilization process, it occurs or it takes place in the ampulla part of the fallopian tube, and it can occur only if the sperm and ovum they are transported simultaneously to the ampullary region. Then only the fertilization will be successful, otherwise there would not be fertilization process. Means their transport should be simultaneous, and if it is not simultaneous then there would not be any fertilization, even there is copulation and no more pregnancy. Now let we consider that the fertilization occurred. If the fertilization occurred, the sperm comes in contact with the membranes of ovum. So outermost membrane is corona radiata, then zona pellucida, and the innermost one is the vitelline membrane, or we can call it as a plasma membrane. So only one of the sperm will fuse with the ovum. So in this process, the acrosome of the sperm is having the acrosome at its tip, a cap-like structure it is, and this acrosome consists of some enzymes and we call them as sperm lysine, and the special or very important enzyme this sperm lysine consists of, and that is hyaluronidase. So this is the enzyme which will help in the dissolution of or dissolving of the membranes of the ovum, that is all three membranes, corona radiata, zona pellucida and the plasma membrane, and this way the sperm will make a path to enter into the cytoplasm of the ovum. So as the sperm enters after dissolving these membranes, it induces some changes in the membranes so that no more or more sperms cannot enter. It blocks the entry of more sperms. As well as along with this, it induces the completion of meiotic division in the secondary oocyte. One thing is very important here. In the previous video, in the oogenesis, while going through the oogenesis, already this is explained in the video that the ovum or the female gamete, we can say female gamete, it is released in the secondary oocyte stage. So means still one more meiotic division is left over here. First of all, meiotic division occurs in the primary spermatocyte, so it leads to secondary meiotic division occurs in the primary oocyte and it leads to the formation of secondary oocyte and a small polar body. Means still the second meiotic division is left. So entry of the sperm in the ovum or in the female gamete leads to the completion of second meiotic division, and this division is also unequal and it leads to the formation of ovum and the second polar body. Means again the two different cells, means different in size they are formed. The ovum is bigger and the polar body is smaller. And after that, the fusion occurs between the nucleus of the ovum and the nucleus of the sperm, and it leads to the formation of zygote. The zygote, when it is start, because this is going on in the fallopian tube, and after the formation of zygote will move towards the uterus, and as it starts moving towards the uterus, at the same time there starts the mitotic division. This will occur, and continuous mitotic division occurs, and this we call as the cleavage. So this is continued, and the zygote divides to form 2, 4, 8 and 16 cells, and all these cells are called as the blastomeres. When the number of cells are 8 to 16, this is called as the morula. And in the next video we will study the morula and blastula in detail. So here morula is the stage when the stage of the embryo having the 8 to 16 cells. And then what happens, there is arrangement of blastomeres, means rearrangement of blastomeres, and they just get arranged along the periphery and leaving a central cavity, and the central cavity is called as the blastocele. And the blastomeres, which are bigger in the size, they just move to one side of the embryo, and that is called as the embryonal knob. So again it divides and give rise to the 32 celled stage, and this is called as the blastula, or we can call this as a blastocyst, as the cells get rearranged. And this is the stage, blastocyst, when the implantation occurs. So implantation, when we say, it means the attachment of this embryo or this blastocyst with the wall of uterus. This is called as implantation. So it is the attachment. So the outermost cells, which forms a continuous layer, that is called as the trophoblast cell, and the inner group of cells is called as the which is attached to this, that will be the embryonal knob. So the trophoblast gets attached with the endometrium, and slowly the uterine cells divide and redivide, and slowly it covers the blastocyst, and the blastocyst becomes embedded in the wall of uterus. The wall is endometrium, which is the innermost layer of the uterus, and this way the implantation occurred. So in this slide there is what is implantation and how does it occur. So this whole video is about the fertilization and the implantation. This is the diagram which is showing the different stages, means this is showing the fertilization process as well as movement of the zygote, or we can call it as an embryo, towards the uterus. So this is given, means from the release of ovum to the implantation. All these stages are shown over here. The different stages are shown as A, B, C, D, E, and this way. Okay, so the implantation of blastocyst is also shown in this diagram. After going through the whole video, you will be able to solve these questions.